up the DNA, they don't cut it right in the middle. They cut it with little ends. That these ends right here where my hand is, they match up. So here's your DNA. Instead of cutting it right here, they cut it so that there's a little bit of DNA sticking. So that once I study this piece of DNA and Derek studies this piece of DNA, we can match up kind of like a puzzle. This region of my DNA that I studied and this region of the DNA that Derek studied, it matches up. So by doing that, they're able to put the whole piece of DNA back in one piece. That's how they did it. So by having this technique, shotgun sequencing, I don't have to do it by myself. I can hire 10,000 labs to do it. 10,000. And that's what the government did. They hired thousands of these labs to analyze from around the world. This was a human genome project, okay? Of all, I mean, this is like nationwide. How much did it cost? Something like $10 billion. That's what they were projecting. How long would it take? 5 to 10 years. That's what they were thinking. That would take 10 years to analyze the human genome because we have so much. Now, if one lab were to do it, it would take like 30, 40, 50 years. But because we have thousands of labs doing it at the same time, and then we have another team that puts it together, right? Once you sequence it, that means you read the sequence, the ATGCs, and you send it back to the labs to put the puzzles together, you're able to determine the order of the nucleotides of the chromosome. And you put it back. So purple goes with purple, and the yellow, and the green, and the red. You see what I mean? And they did the same for all 46 chromosomes. All of them. All of them. All of them. Now, how long did that take? Right? How long did it really take? Okay? So, they started this, people, in 1990. They started the Human Genome Project in 1990. They were projecting 20, 30 years, right? 20, 30 years, guys. It's a long time, right? It did not take 20, 30 years. This is when they finished. They finished in 2003. That's 13 years. It took 13 years. It's a long time. Now, did they use computers in 1990? No, they used just manual labor. So it took so long. Nowadays, we use computers, okay? And so it took 13 years, $2 billion, cost $2 billion to analyze it. And so now in 2003, an international effort known as the Human Genome Project finished the first human DNA sequence. Now, what did we learn from it? 4 things. Number 1, we learned that which region of the DNA transcribes RNA, makes RNA. Remember the 3 types of RNA? We learned that our DNA is divided up into regions. Not all of our DNA represents a gene. Only a third of it represents a gene. Now, how many genes do we have? This is how many genes we have. 21,000. Humans have 21,000 genes. And the 21,000 genes only make up 30% of the DNA. What about the other 30%? 30% or the 33%, okay, I should make it, okay, to make it equal to 100. Some mathematicians in here would tell me, okay? So 33% would be for genes. The other 33% would be for control, gene expression, control. Those are regions where you make RNAs, you make tags, you control how genes are expressed. And what about the other 33% of the DNA? We call that junk. It's just junk DNA. So humans, we have a lot of junk in our DNA. DNA that doesn't mean anything. See, it doesn't mean anything. It's just there. So this is what they found. They found that here, if this represents 100%, this is our DNA. This is how much we don't know yet, okay? We're still studying. These regions here represents each of these activity. This is what they make inside your cells. So 33% of a cell's DNA is for genes. 33% is for gene regulation. 33% of our DNA is just junk, 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 junk. Does nothing. Yeah, guys, good so far? Good so far? Okay, the story continues. So then once they successfully sequence the human genome, all 6 billion nucleotides, all of it, and they study each one of the chromosome, they found that the human genome had 21,000 genes, right? What's the next step? They started sequencing other species. They started to sequence bacteria, chickens. Humans, we did it already. Water fleas, rice, plants, flowers, roses, chimpanzees, orangutan, every single species. They sequence every single species. And then now what next? What do you do next? Now they have a database, a system, a machine that saves all of these DNA sequence, the order of the ATGCs. So this is what they discovered. They discovered that if you compare the DNA of human to human, we are 99.9% the same. So if you compare two humans, right? Two humans. So I compare my DNA to, let's say, Bhavisha's DNA. Bhavisha and my DNA. We are 99.99% similar. Bhavisha, you and I are almost cousins, or brothers, sisters, or maybe dad, daughter. Bhavisha, you hear what I said? She okay? Bhavisha, are you okay? I only see her forehead. All right. So let's say Alex. Okay, Alex, you and I are 99.999% similar in DNA. There's very little difference. But because of the Human Genome Project, they estimate that every 1,200 bases that you and I have, there's only one base that's different. One base. So out of 1,200 bases that you and I are similar, one of them, one letter, ATGC, is different. One out of 1,200. That's very small, right? It's very tiny. And so, what makes it different? What's that one? What's this? What's up with this guy? How come? What's the difference? What makes it different? The difference is this one base that's different between myself and Alex and I is, number one, our traits. He might have brown hair, I have black hair. He has a round chin, I have a cleft chin. You see what I mean? That one base makes us different. And then this is what they also found. Remember that 33% junk DNA region? Remember that 33%, guys? 33% junk DNA, junk. I say it junk because it doesn't represent junk, okay? Junk DNA, which is, it doesn't represent a gene or anything, nothing. In that region, when they compare humans to human, they discovered that inside the junk DNA region, there's a difference between humans. They found that there's this region called single nucleotide polymorphisms. They found these single nucleotide polymorphisms in the junk region. And they wonder, the heck? Why is it different? Why does it repeat? Why is it different between different species? This is what they discovered. Because they believe that humans descended from other species. So, the most ancient animal, the most ancient species on Earth is your blue-green algae. We, you and I, we have blue-green algae DNA inside of us, inside the junk. Our oldest animal cousin is I think it's a lancelet or a lamprey, like earthworms. Earthworms, we have earthworm DNA. Is it functional? It's in the junk. You guys got it? We have monkey DNA. We have monkey DNA. Is it functional? Not functional. It's in the junk DNA. So when you compare humans to humans, like let's say brothers and brothers, two brothers, right, from the same family, they found that they have very similar single nucleotide polymorphisms, very similar. But when you compare brother to cousin, there is slight difference in their polymorphisms. And then if you compare cousins, or you to, let's say, your third generation cousin, it's even slightly more different. So if you compare yours, Nathan, to mine, you and I are even slightly more different. You guys see where I'm going? So if you were to do a DNA test, paternity test, so let's say, I'll give you an example. Let's say you have this boy, me, okay? And I was abandoned by my parents when I was a baby. And they left me at an orphanage. And later on in life, my parents wanted to find me, okay? Because I guess they turned their life around. But they don't know if I'm the son or not. So what they do is they take samples of my DNA and they take samples of their DNA. So the mom and the dad, supposedly my parents. They will compare the single nucleotide polymorphisms with mine, and it should match up. That's how they do it. Okay? That's how you do it. Now, how come I don't compare like pieces of DNA like hair? Because look in the Zoom. How many people have dark hair? Right? Everyone. So you can't use genes like hair to determine paternity test. You cannot use, let's say, how many of you in here, how many do you see have two eyebrows? How many people? 95% of us? Who plucks their eyebrows? Who plucks their eyebrows? Come on, be honest. Who has a unibrow in here? Everyone has two eyebrows, right? So that means that can you use the eyebrow gene to compare to your parents to see if you're the son or daughter? You can't do that, right? You cannot use genes to determine paternity or maternity. Okay? So therefore you have to use something else. You have to use regions of the DNA where it is only uniquely you. So therefore, what do? You go after single nucleotide polymorphisms. Okay, guys, does that make sense? Make sense? All right. Okay, you cannot use skin tone. You cannot use hair color. You cannot use eye color, eye shape, chin, ears. You can't, because so many people could have it, right? You can't. If I do something like that, everyone's my kid, right? You're all my kids. You see what I mean? Because we are 99.99% the same. So I use genes. Well, David's my son. You see what I mean? Nathan's my son. That doesn't make sense. All right. So this region, which makes up about 1/3 of your DNA, it's just junk. But we found use for them because we're able to identify the identity of a child to a parent. Second thing is, this region also has a unique property, such as you can determine if a person will have a particular disease or a certain medical condition. Because this region, although I use the word junk, sometimes certain regions of this DNA, it gets activated. And when it gets activated, it goes to that region, it goes to the gene region, and it activates a protein. That protein goes to a gene that causes cancer, and you get cancer. Guys, does that make sense? So scientists have learned that just because a person smokes cigarette, 40 years of life, 40 years, will that person get lung cancer? No. But the person is susceptible to lung cancer more than somebody who doesn't smoke, right? But it doesn't mean that you will get lung cancer if you smoke cigarettes. Or Would you get skin cancer if you're in the sun a lot? No, it doesn't mean that. But it makes you more likely to get skin cancer if you're in the sun. So certain people could get skin cancer much easier than others because their single nucleotide polymorphisms, DNA, it gets activated and it triggers a certain disease to become active, and then you get it. Got it? Now, remember how I said that cancer, all of us hopefully have known somebody who has cancer, right? And so how does that person get cancer? Now, even if they're in the same family, if your sister gets cancer, does it mean that you get cancer because you're the same DNA? It's your sister. No, it doesn't mean that, okay? It could mean a variety of things. And one of those things is this DNA region in your cells that could activate because you trigger it by smoking, by exposure to certain chemicals, by exposure to the sun, things like that. Now, so in 2008, as they learned more about DNA in 2008, for fear of abuse of the genetic knowledge, the government created a law or an act, a law. Act is the same thing as law. They call it Genetic Information Non-Discrimination Act. And what it does is it prevents insurance companies and employers from discriminating against you if they have access to your DNA. Now, if they have access to your DNA, insurance companies would know if you are likely to get heart disease or not. They would know if you have diabetes, type 2. They would know if you are going to develop some autoimmune disease when you're 40 years old. You see what I mean? So they cannot discriminate. Now, is there a DNA bank where they put all the everyone's DNA? The answer is yes. So every time a person is convicted of a crime and goes to jail, they take DNA samples, they put it in. Because you're a criminal, you lost your rights. Now, in the past, what they do, even still now, they do fingerprints, right? Fingerprints. So for me to do this job, I had before I started teaching, they do a fingerprint of me. So they put my fingerprints in a system, in a bank. And then this computer generates a search on my background to see if there's something wrong with me, mental problems, felony, misdemeanor, any arrest, things like that, drugs, things like that. So they want to make sure that I'm legit, that I'm clean before they let me be a teacher. Yeah, you guys make sense? So there is a system, a bank available that the government have access to. So, but this law, it prevents companies and the government from discriminating against you just because you have these genetic defects. Okay. Now, one of those things that you can also find on these sites, these computer bank, these DNA bank, is called genomic imprinting. Genomic imprinting are little molecules, compounds that are attached to your DNA, and it causes your DNA to change. And so by analyzing DNA, the DNA, you're able to come up with the kind of like a review, like kind of like a big picture of a person's profile, genetic profile. Kind of like this. Right here, this is called genomic imprinting. Right here. This is called genomic imprinting. This is called methylation, okay? Well, this is like AP, okay? This is called methylation. Methylation is the process of adding a certain compound to your DNA so that you can deactivate it or activate it, okay? So that's called genomic imprinting, okay? Genomic imprinting, okay? And that, people, is less than